Hello students, welcome to Rayat Online School Education Project conducted by Rayat Shikshan Sanstha Satara, Sadguru Gadge Maharaj College Karad, conducted here 12th Rose Project 2021 to 2022. Myself, Miss Mohite P.S. My subject is biology and my topic is enhancement of food production. This is the second lecture of enhancement of food production. In this lecture, we see tissue culture. Tissue culture, it is actually a collection of different techniques. Tissue culture is the growth of tissue or cells in an artificial medium separate from the parent organism. This technique is also called as micropropagation. Here, tissue culture is a technique of plant biotechnology in which here isolated cells, tissues, organs are grown in vitro on a solid or liquid nutrient medium under aseptic and controlled conditions of light, humidity, and temperature for achieving different objectives. The part of a plant used in tissue culture is called explant. Here we see plant tissue culture is based on principle of totipotency. Here totipotency means which is an inherent ability of a living plant cell to grow, divide, redivide, and give rise to a whole plant. The plant tissue culture medium contains all essential minerals, sources for carbohydrates, proteins, and fats, water, growth hormones, vitamins, and agar for callus culture. The most commonly preferred medium for tissue culture is MS, that is the Murashic, and Skug medium. Here we see the part of plant. The part of plant used in tissue culture is called explant, and the most commonly preferred medium for tissue culture is MS medium, that is the Murashic and Skug medium. Based on the nature of explant, tissue culture may be, there are three types, which are cell culture, organ culture, and embryo culture. Based on the type of in vitro growth, there are two types, which are callus culture, and that is callus culture, that is the solid medium, and suspension culture, that is the liquid medium. Here in tissue culture, maintenance of aseptic conditions. aseptic condition is essential so as to avoid contamination by undesired microorganisms. It is necessary to sterilize. It is accomplished by sterilization of glassware. Here, sterilization of glassware here used of detergents and a hot air oven. Next sterilization is nutrient medium. Nutrient medium sterilized by autoclave under constant pressure of 15 lb per sq inch for continuous 20 minutes. Next sterilization, that is the explant. Here by treatment of 20% ethyl alcohol and 0.1% of HgCl2 used for sterilization of explant. Next sterilization is inoculation chamber. Inoculation chamber sterilized by laminar air flow by using UV ray tube for 1 hour before performing actual inoculation of explant on the sterilized nutrient medium. In callus culture, the solid medium is used. The development and organization of tissue is lost. Hence, the cells of explants divide and redivide to form a mass of undifferentiated cells called a callus. It is maintained on a solid medium. Here, callus culture uses solid medium. Here we see diagram, that is the steps in plant tissue culture, that is the callus culture. Here we see mother plant, here in test tube, explants, then formation of callus, and then formation of shoot and root, that is the organogenesis. Here organs are produced. Shoot and roots are the organs of a plant body, and then multiple plants are here produced, that is hardening. Here we see callus can be induced to form roots and shoots. In suspension culture, small groups of cells or a single cell are used as explant in the liquid medium. The liquid medium is constantly agitated by using shakers, that is the agitators, so that there is a constant mixing of a medium and the explant. Both the callus and suspension culture die in due course of time. Therefore, subculturing is necessary for a continuation of the technique. Both the callus and suspension cultures need periodically subculturing. Micropropagation, that is the clonal propagation, organogenesis via shoots is considered as one of the most widely used commercial method of regeneration of a plant. In suspension culture, small groups of cells or a single cell are used as a explant in the liquid medium. The liquid medium is constantly agitated. Here we see micropropagation, that is the clonal propagation. Organogenesis via shoot is considered as one of the most widely used commercial method of regeneration of a plant. Here micropropagation is also known as a clonal propagation. Micropropagation helps in a rapid multiplication in plants. Micropropagation helps in a rapid multiplication in plants. It can produce plant throughout the year independent of seasons. It produces genetically similar plants, that is called as clones. The rare plant and endangered species are multiplied by this method and such plants are saved. Somatic hybrid can be produced in short time span. Here we see a flow chart of a tissue culture technique. In this chart, first step we see here cleaning of glassware, sterilization of glassware and instrument in an oven or a autoclave. Next step is a selection and preparation of a nutrient medium, that is MS medium, with a known concentration and proportion of different components. Sterilization of medium in an autoclave for a continuous 20 minutes under constant pressure of 15 lb per inches. Next step is a preparation of a plant material, that is the explant, includes isolation of explant followed by surface sterilization and rinsing with water. Explant is obtained from the growing stock plant. Next step is a inoculation of the explant in the culture flask containing sterilized nutrient medium. Inoculation is done in the laminar airflow cabinet unit. Next step is a inoculation of the inoculated explant. Here cells of explant grow, proliferate to form callus within 2-3 weeks. Next step is a subculturing of the callus. That is, if callus is to be maintained for longer period, callus is divided into 3-4 segments and then transferred to fresh culture medium. Next step is organogenesis. Here, initiation of rooting and shooting that eventually leads to plantlet formation. Next step is hardening. Hardening. In this step, plantlets are transferred to polythene bags containing sterilized soil and kept at low light and high humid conditions for suitable period of time. And last step is transferred to field. Next slide is advantages of micropropagation. Your first advantage is it helps in rapid multiplication of plant. Second advantage is a large number of plantlets are obtained within a short period and from a small space. Third advantage is plants are obtained throughout the year under controlled conditions independent of seasons. Fourth is a genetically similar plants, that is the clones, are produced or formed by this method. Therefore, desirable characters, that is a genotype, and desired sex of a superior variety are kept constant for many generations. Next advantage is the rare plant and endangered species are multiplied by this method and such plants are saved. Go to next slide. Here we see next point, that is the single cell protein, that is the SCP. In this point, see here, by 2050 the world would need to produce 1250 million tons of meat and dairy products per year to meet global demand for animal derived protein at current consumption level. Moreover, human population is underdeveloped and even in the developing countries is suffering from protein malnutrition, resulting into variety of nutritional diseases. To fight with these, efforts are undertaken by conventional methods to increase the food yield by different methods of crop improvement, use of biofertilizers, biopesticides, chemical fertilizers, and high yielding varieties, that is the green revolution. Here, single cell protein refer to the crude or a refined edible protein extracted from pure microbial cultures or from dead or dried cell biomass. Here, SCP, that is the single cell protein, is also rich in vitamins, vitamin B complex, minerals, and fats. Here, microorganisms like algae, fungi, yeast, and bacteria have very high protein content in their biomass. Here we see substrate used. Here, agricultural wastes, wood shavings, sawdust, corn cobs, paraffin, N-alkanes, sugarcane molasses, even human and animal waste are used for the single cell protein. Then the high quality proteins produced by microorganisms like algae, fungi can be used as a supplement in human and animal feed. The single cell protein can be readily used as a fodder for achieving fattening of calves, pigs, in breeding fish, and even in poultry and cattle farming. Here, the microorganisms used for the production of SCP. Some examples I will tell you. Fungi, Aspergillus niger, Trichoderma viride. In yeast, Saccharomyces cerevisiae, Candida utilis. In algae, Spirulina species, Chlorella pyrenoidesa. bacteria, Methylophilus, Methylotrophus, Bacillus megasterium. In the next slide, we see advantages of a single cell protein. First advantage is microorganisms have a high rate of multiplication. That means a large quantity of biomass can be produced in a comparatively short duration. Second advantage is the microbes can be easily genetically modified to vary the amino acid composition. Third advantage is a broad variety of raw materials, including waste materials, can be used as a substrate for SCP. This also helps in decreasing the number of pollutants. SCP, that is the single cell protein, serves as a good source of vitamins, amino acids, minerals, crude fibers, etc. Go to next slide. Here, next point is a biofortification. Biofortification is the method in which crops are bred for having higher levels of vitamins, minerals, and fats, that is the better nutritive value. Due to this, problem of malnutrition can be overcome. Following objectives were considered for a breeding program: protein content and quality, oil content and quality, vitamin contents, then micronutrient content and quality. Go to next slide. It is a focus on making plant food more nutritive as a plant grow or develop. Here, biofortification can be achieved through conventional selective breeding practices and through rDNA technology. Here we see some example of a biofortification, that is the fortified maize having twice the amount of amino acids, that is the lysine and tryptophan. Second example is a wheat Atlas 66 has a higher protein content and iron. Fortified rice has a 5 times more iron are developed. Third example is a vegetable crops like carrot and spinach have more vitamin A and mineral. Here our second lecture is finished. Thank you.